Welcome to today's video on experimental method of data collection part 2. Today we will focus on experimental designs. A quick recap. A good experimental design is necessary to reduce the non-sampling errors. For a good experimental design, the three criteria laid down by R.A. Fisher, replication, randomization and local control have to be incorporated in the design. It is also necessary to include a control group in the design of the experiment. We have seen about replication. Today we will see about randomization. Randomization ensures that equal chance is provided to every treatment for being assigned to any experimental unit. Treatment, we have seen about this term in our previous video. It is an independent variable which the experimenter will manipulate depending on the type of study. For example, if it is a study on the effect of pollutants on plant growth, exposure to pollutants will be the treatment. Experimental unit in an in vitro study may be a test tube or a Petri plate. In an in vivo study, it may be a pot or plot in which a plant is grown, a laboratory animal or a human subject depending upon the research investigation. The treatments have to be randomly allotted to the experimental units. This may be done by employing the lottery method or the random number table method. By randomly allotting the treatment to different experimental units, we will be able to get an unbiased estimate of differences between the treatment. When randomization is done along with replication, it decreases the error associated with the difference in treatment. We will see a few important experimental designs. The first one is completely randomized design. As the name itself implies, the treatments are assigned completely at random to the experimental units without taking any other criteria into consideration. Completely randomized design is appropriate for experiments with homogeneous experimental units or for laboratory experiments where the environmental effects are easy to control and when we do not anticipate any source of variation. Let us consider that we have three treatments A, B and C and we have 15 experimental units which we have shown as squares. This figure shows how we have randomly allotted the treatments to different experimental units. 5 experimental units will receive treatment A, 5 will receive treatment B, 5 will receive treatment C. So the element of replication can also be incorporated. If the experimental units are more, replication also increases. In all cases, it is not possible for us to go for a completely randomized design. When we are expecting a source of variation or a source of variation is existing, then to eliminate the error due to the source of variation, we have to go to randomized block design. A source of variation may arise if we are not able to do the experiment at the same site or at the same time, or when there is a scarcity of resources, or when the experimental units are not homogeneous. Let us see with examples. Suppose we are doing a study on plant growth and we have three treatments A, B and C. So we have to do all the treatments with replication. But we don't have one field where all the treatments can be done with replication. So we have to do the experiment in three fields. So we have to consider each of these fields as a block. This is done to eliminate the error that may arise because of differences in the fields. So each field has to be considered as a block and all the treatments, for us we have three treatments. So all the three treatments have to be done in each of these blocks. By doing this, we are eliminating any error that may arise because of field differences. The treatments are randomly assigned to the blocks, so that is why it's called randomized block design. Whenever any source of variation arises, we have to always ensure that it is among the blocks and not within the block. Let us see with an example. Suppose we are not able to take the readings for all the plant parameters on a single day. Then it has to be ensured that one block is completed per day. By doing the measurements for all the treatments of one block, we eliminate any day differences that may arise between the treatment groups. The next block can be completed on another day. Similarly, if we have more than one observer who is going to take the reading for one block fully has to be done by one observer so that we eliminate any error that may arise out of observer differences between the treatment groups. Another observer has to do for full block 2. So, randomized block design helps us to isolate and reduce any source of variation that may arise. Let us see with another example. Suppose we are doing a in vitro study where we have 4 treatments, but we don't have one incubator where all the plates of the treatments with replicates can be kept. And so we have to do in 2 incubators. Then each of these incubator has to be considered as a block, and the plates of all the treatments have to be placed in both of these blocks. This is an attempt to reduce incubator differences. When we do this randomized block design itself, we are able to bring in the element of replication, whereas it is not sufficient in some cases. When we need to increase the replication, if possible, we can do the replication within a block. So here, when we are keeping the treatment plates, we can keep 2 or 3 plates of treatment B, 2 or 3 plates of treatment C, similarly A and D. How many ever plates we are keeping, same we have to keep for all the treatments. By doing this, we will be able to increase the number of replication. If it is not possible to do a replication within a block due to constraints, if we are repeating the experiment at a later time, again we have to remember the concept of the block, and the whole block of experiments have to be repeated together for replication. Randomized block design is suitable for field experiments where the external factors are difficult to control. But when the number of treatments is large, adopting a randomized block design may be difficult. As is evident, in randomized block design, we are able to eliminate the error that may arise due to one source of variation. But when we have to simultaneously remove the errors due to 2 sources of variations, then we have to go for Latin square design. Here, one source of variation is given as row and another source of variation is given as column. And depending upon the number of treatments, you will have rows and columns. If you have 3 treatments, we will have 3 rows and 3 columns. This resembles a square, and initially the treatments used to be represented as Latin letters, so it's called as Latin square design. Let us now see the examples. Suppose we have to do an experiment with plants on a field of land that is a slope. So the slope may contribute to a variation. And there is also a gradient of soil texture which can also contribute to variation. So simultaneously we have to eliminate 2 sources of variation. So here as rows we have put the slope, we have classified into highest part, mid part and lowest part of the slope, and the soil texture again we have been able to put it as grade 1, grade 2 and grade 3. Now, we have to follow a design where each of these treatment occurs once in each row and in each column. By doing this, we ensure that, for example, treatment A is done in the highest part of the slope, mid part of the slope as well as the lowest part of the slope. Similarly, it has been done with the soil texture grade 1, soil texture grade 2 as well as soil texture grade 3. So, we are able to eliminate 2 sources of variation. Each treatment appearing once in each row and each column makes one row as a block and one column as a block. Thereby, we are able to eliminate 2 sources of variation. If we see treatments B and C, they also will occur once in each row and once in each column. Let us see another example. Suppose we have 4 diet treatments, but we have only 4 animals for the experiment. So now each of these 4 diet treatments have to be given to all the 4 animals. Only then we will be able to eliminate animal differences. But suppose the duration of the diet treatment is 1 week, then it will take a month for us to complete the study. So there may be variation due to week differences also. So we have put week differences as rows and animals as columns, and we have allotted the treatments in such a way that each treatment appears once in each row and once in each column. So for example, A treatment is done in week 1, week 2, week 3 as well as week 4, and A treatment is also done for animal 1, animal 2, animal 3 as well as animal 4. And that is how we are able to eliminate 2 sources of variation. For each of these treatments, it will be true. For the sake of simplicity, we have followed a pattern in the rows and columns, but the row and column may be randomized, but we have to ensure that each treatment appears once in each row and in each column. So we have seen about randomization. In the upcoming video, we'll see about local controls and how to reduce measurement errors. Thank you.